are live.
Okay. Well, hello everyone. This is an introduction and welcome to our webinar. I'm speaking to you from the United Kingdom. My name is David Galloway. I'm the president of the Centre for Intelligent Design in the UK. And this is going to be the first of 3 webinars that we plan to host during this month of July. We are a network of scientists and academics who are keen to explore the question of whether the apparent design in the universe and in life may actually be genuine rather than illusory. We'll add our web details to the show notes for each webinar, so you'll have a chance to follow up with some of our material if you wish. Now, today we're delighted to have Dr. Mike Behe as our guest speaker, and he'll be providing some insights shortly into the biochemical basis for COVID-19. The COVID-19 pandemic continues to be an urgent problem and presents many unsolved challenges. As of this week, worldwide, there have been some 13.4 million cases confirmed, and some 574,800 people have died. In April of this year, more than half of the world's population lived in countries enforcing a lockdown, resulting in a huge disruption to individuals, to businesses, to entire sectors of society, such as global tourism and travel. Even countries that have suppressed the pandemic are experiencing the harsh economic spillover effects from the rest of the world. Now, actually, this pandemic marks the 3rd deadly outbreak due to a coronavirus, after the SARS syndrome in 2003 and the Middle East respiratory syndrome in 2012. But as you may hear this evening, coronaviruses have been around for a lot longer than that. Now, our 3 webinars are going to be successively from a biochemist this evening, a molecular biologist, and then a public health physician. And the next 2 I will remind you about later, but they're going to be on the next 2 successive Thursdays. After each webinar, there will be some time for questions and discussion. So please add your questions to the YouTube chat facility and try and keep them focused and brief if possible. And Mike will be joined by David Williams and Alistair Noble, and David will be moderating the question session after Mike's talk. So I'm really delighted to welcome our first speaker this evening, Mike Behe. He is a professor of biochemistry at Lehigh University in Pennsylvania and is a senior fellow at the Discovery Institute. Mike came to international prominence in the 1990s on the publication of his book, Darwin's Black Box, where he outlined his views on the irreducible complexity of many biological systems. And he's become somewhat notorious, attacked with vehemence on the internet, questioned, ridiculed. If you find him on Wikipedia, his entry is less than fulsome in its praise for his achievements. Despite that, he maintains a calm confidence that his views are correct. And while many have opposed him, his views on irreducible complexity and on the limits of Darwinian mechanisms to explain life's complexity have not been refuted. He's written subsequent books, The Edge of Evolution in 2007 and Darwin Devolves last year, and these have further elaborated his ideas. So after Mike speaks, I'll invite David Williams to take over the question and discussion session, and we'll aim to conclude this webinar somewhere between an hour and a quarter from now. So now it's a great pleasure to welcome Mike back to speak under the banner of the Centre for Intelligent Design in the UK. He toured the UK and gave numerous lectures about a decade ago, and so I'm very pleased now to ask him to give his talk, The Biochemical Basis of COVID-19. Thank you, Mike.
Thank you very much, Dr. Galloway, for that nice introduction. And it's always great to be able to speak to folks in the United Kingdom. And so I'm grateful to the Center for Intelligent Design for providing me that opportunity. Okay, so I'm going to share my screen with some PowerPoint slides. You've probably never seen PowerPoint slides before, but here we go. Let's see, go up to the top and this one, and oops, did I do that right? Oops, sorry, just getting there. There we go. Okay. All right, that was easy. Okay, so as you can see, this is a series called Design in Virology ? And I'm going to be talking about the biochemical basis of COVID-19. Biochemistry is the molecular basis of life. So it's really where the rubber meets the road, where the real action is. As a biochemist, that's what I say. But I'm going to do a little historical introduction to this topic, just because I think it'll help set the framework for understanding what's going on. And so let's, let's take a look at this slide. It turns out that plagues and viruses and stuff have been around human civilization for a very, very long time. One of the biggest ones was called the Antonine Plague, and it occurred when Marcus Aurelius was in power in Rome. And it's said to have swept through Rome and killed maybe a quarter or a third of the population. So much, much worse than we so far have had it. This is a painting by a later French painter, a 19th century French painter, and he imagines what the inhabitants of Rome must have thought. They see everybody dropping down and dying and they have no idea what's going on. They can't see anything. They just see people starting to die. And for all they know, it might have been, it might have been the angel of death centered here in the picture. So it was doubly terrifying. Not only were people dying, nobody had even the slightest clue as to what this is. Later historians think that it might have been smallpox or measles, but whatever it was very deadly. But science advances with new technology. Without technology, we're stuck with our own senses, our eyes and ears and so on, and they have limits. They can't see below a certain point. So the big advance, the big breakthrough for biology was with the invention of the microscope in the 17th century. The microscope allowed people to see things that were too small to see with the unaided eye. And when this was first used, even this crude microscope revealed things that nobody knew existed. For example, here's a drawing by the microscopist Robert Hooke, and he just looked at insects, and he saw in them, he saw all sorts of strange structures, all sorts of detail that nobody had ever seen before. If you look at the bee in the lower right-hand figure, you can see the compound eyes of the insect. Nobody knew that insects had such features. Now, it seemed like something from an alien world. Let's see here. Oop, going the wrong way. Oh, I'm sorry, let me go back once. And if you look at the top, you can see a flea here and these two tiny mites that actually were on the flea. And this led the satirist Jonathan Swift, who, as you know, wrote Gulliver's Travels and other works. He made up a little poem. He wrote, "So naturalists observe a flea, has smaller fleas that on him prey, and these have smaller still to bite 'em, and so proceed ad infinitum." So the idea was that this flea that we just saw the details of has even smaller fleas, and perhaps they have smaller fleas and they have smaller fleas. The point is that microscope revealed that there was a hidden world. There were details to life beyond imagining. These were no longer the familiar animals of field and forest. These were in many ways very alien. And Swift imagined that maybe these fleas just went on to infinity. We know now that that's not the case, but nonetheless, you can sympathize with him and others of the time that, you know, things might go on to a very, very, very deep level of which they were unaware. And the historian Charles Singer wrote of this time period that the infinite complexity of living things thus revealed was as philosophically disturbing as the ordered majesty of the astronomical world, which Galileo had unveiled to the previous generation. So in both cases, they seemed to discover that there was a whole lot more to this universe and to life than people had previously supposed. Well, if science advances with technology, then if you improve the technology, it can advance even more. And the first crude microscopes were eventually improved and then improved some more by a variety of techniques. One of the, in the 19th century, there were new, better lenses, and there was oil immersion, and there was staining to stain parts of a cell. But the big thing was the discovery of electricity, and that allowed electrically powered instruments to be used. And that was nice because now you could have an electric light illuminating the sample, whereas before people had to rely on natural light. Not only that, but electricity allowed for the invention of something called the electron microscope, which uses electrons instead of light to try to illuminate a sample. And you have to, and this is continuing. The improvement of techniques is continuing apace, and we know much, much more than we did just 20, 30 years ago. And to appreciate the surprise and the philosophical unease that people felt in the 17th century, here's a result from some new microscopic and videographic techniques that have come up in our own time. This is taken from a paper in the journal Science, and it's an image of this ordinary insect, a plant hopper, a very common one. And people had known for a long time that the plant hopper had bumps on its hind legs at one stage of development, but they could never figure out what they were, and they couldn't really see them too closely. But these British scientists figured out a way to improve their optical techniques, and they saw that those bumps were actually gears, real mechanical interdigitating gears. And if you followed it like I did, on the internet was a buzz for weeks on the philosophical and even theological ramifications of this discovery of these things. So here's a little picture showing different levels of life. From the human, this is a little figure of me when I was postdoc, and then this is skin and tissue and a cell and a subcellular organelle called mitochondria and the membrane of that, and then a molecule, a protein that is in the membrane of that. And for each level, for each arrow, the size decreases about 30-fold. So that overall, from the man down to the molecule, it's a range of about a billion. or 10 to the ninth. I forgot. I think the British and Americans disagree on what a billion versus a trillion is, but 10 to the ninth. And it turns out that this down here is where the machinery of life is. And of course, life needs a lot of machines to work. So it turns out that viruses and bacteria inhabit the range around here or so. Whereas in Roman times, they were confined to this level. And with the first microscope, somewhere around here, we have gotten all the way down to the very basic level of life. And at each stage, we've been astounded repeatedly on what we have discovered. So let's concentrate down here and ask ourselves, what's the structure of the viruses? The viruses, we'll see, are made of molecules, these smaller, and the molecules, the proteins, the protein machinery is part of the makeup of the virus. In order to introduce you to this topic, I'm not going to talk about COVID first. First, I'm going to use a different virus as an introduction. This here is Bacteriophage T4. A bacteriophage is a virus that infects bacteria. And just like with people and other organisms, the virus infects it, sends its DNA into it, and uses the cell to make copies of itself. So on the left, you see an electron micrograph of the bacteriophage, and not very good resolution, but here you see a drawing of it using many images to get a better picture of what we're looking at. And as you can see, this is a weird looking thing. It looks like a lunar lander or some kind of machine. And it looks like some kind of machine because that's what it is. In recent years, an even newer technique of microscopy has been invented called cryo-electron microscopy. And cryo-electron microscopy has given us these very, very detailed pictures of portions of the bacteriophage. This is a bacteriophage as it exists in solution. It hasn't yet attached to a cell. And this is the big long shaft you might remember from the previous picture. And you see all these little dots here. You probably can't really see them too well because of your resolution in your screen. But each of these tiny fuzzy little dot things is an atom or a group of atoms, a small group of atoms. And this is composed of molecules, protein molecules that make up the shaft here. And so is this area and down here too. This is an assembly of molecules. And when this portion down here touches the cell, it springs into action, and the legs expand, and this shaft contracts. And as it contracts, it pushes this needle-like tube into the cell. It pierces the membrane and pushes it into the cell and injects its DNA into the cell. And the DNA then reprograms the cell and makes more copies of this bacteriophage. Now, to reinforce things, I want to show you, here's the drawing of the bacteriophage T4. And scientists have focused on this area called the base plate and gotten really good pictures of this. And they've gotten it in a number of states and have used that to make a movie of what happens when it touches a cell. And here it is right here. This is how it starts. And when it touches a cell, the legs expand, and then this will, the shaft will contract and inject the cell. So I just want to show this from a recent paper because it shows you that these things are machines. I mean, they're enormously complex molecular machines. All of these dots and so on, they're all made out of protein molecules, groups of atoms, small groups of atoms. Okay, well, now that doesn't look like the virus that we're talking about today, the virus of interest today. COVID-19 doesn't have that real mechanical look that Bacteriophage T4 does, but that's kind of a mistake just because it uses different mechanical means to propagate. COVID, of course, looks kind of like a soccer ball with spikes coming out of it. And instead of injecting its RNA into the cell, and of course, the COVID virus has an RNA genome rather than DNA, but that's okay. We'll overlook that for our purposes here. The COVID virus here attaches to a protein machine, which we can call a receptor, on the surface of the cell. Now, the cell, eukaryotic cells, cells that have nuclei, have machinery that when something attaches to specific receptors, then the cell thinks that's something that it should take into its insides, because organisms communicate between cells, and so cells need some method to take in foodstuffs or messages or other things from the outside. Now, these things here, these receptors, they are not there simply for the purpose of binding the virus. The virus uses them. They usually bind to something else that helps the organism survive and live. But when the virus tricks them into thinking that they should take it in, it is pulled inside the cell by machinery that's already in the cell. And you can see the nucleic acid shown here as a little squiggly line, and the residue of the virus here. And once inside, the nucleic acid hijacks the cell machinery and tells it to make copies of the components of the COVID virus. And after a while, the COVID viruses break free and go out of the cell, and it oftentimes leaves the cell dead and trashed and dying. Okay, so why does this stick to here? Why doesn't it just go bouncing around and bounce here like it might bounce off a rock or something? Well, it turns out that proteins, this is a detailed structure of a part of a protein. All these little ball-like things are atoms. And here's a second protein. The second protein binds to the first, and when it binds, the two surfaces come together, and they are complementary in both a geometrical and a chemical sense. And that's seen better on the next slide. Here's a little cartoon of two proteins binding. This is one protein, and this green thing here with a depression in the middle is supposed to be a second larger protein. The idea is that this one comes down in and binds to the first one. And notice that the shapes are geometrically complementary to each other, but that's not the only thing. Notice here that there's a negative charge next to a positive charge here. Turns out proteins can have electrically charged groups, negative and positive, and when they are arranged so that they're next to each other, that's an attractive force that helps the proteins bind to each other. And these little things are also chemically complementary. These are oily patches. So an oily patch on the protein comes and nestles next to an oily patch on the second protein. And there are other kinds of interactions which chemically attract this protein to this protein and allow the complex to stick together. These are, you can think of them as kind of like tiny magnets at these various places on the surfaces that allow these things to stick together. Now, all proteins work by binding stuff, by binding other proteins or other molecules. And here's a couple of analogies that you can use to kind of understand the concept. One protein molecule might be like a big puzzle that has a piece missing. And the smaller molecule that it binds might be like that, the piece that has the missing shape. So it can stick in here and sticks pretty well because it fits firmly inside. Another analogy is a lock and a key. The protein, the big molecule, is like the lock, and it's got a place where a key can fit, but the key has to have the correct structure in order to fit in, or else it simply does not work. Now, what does this have to do with viruses? Well, it turns out that viruses have to get into cells in order to hijack them. But cells don't want viruses to come in. So essentially, the viruses have to act as burglars and use tools to get into the cell. And if you look, one kind of tool is shown here, the crowbar. Crowbar is a tool where you can force a door open. You break down the door. And that's kind of analogous to Bacteriophage T4, which kind of jams its needle-like structure into the cell and forces its DNA inside. But COVID does not have structures like that. And whoops, I'm sorry. Instead, it has structure more akin to skeleton keys that a burglar might carry. That is, they have a shape that might fit some lock. And if the COVID can unlock the door and get inside, then it can use other standard tools to do its nasty work inside. Okay, well, one interesting thing is that if you read up on this story, scientists have said, well, it seems that this COVID strain, the COVID-19, mutated and developed the ability to get to infect humans. Well, how does that happen? What does it take to mutate a virus so that it can infect a human cell? Well, let's do a little bit of homework first and ask yourself, what kinds of mutations can arise in DNA or RNA as well, but we'll stick with DNA here. Turns out that there are different kinds of events that are all listed as mutations, and they all have in common that they make a change in the structure of a DNA, specifically in its sequence of subunits called nucleotides. And the simplest kind of mutation is called a substitution, where one unit, one nucleotide in DNA can be switched for another kind of nucleotide. This is not done on purpose. Rather, when DNA is replicated, mistakes can be made. It's a very accurate process, but occasionally some mistakes are made, and that can result in what's called a substitution mutation. Another kind is called a deletion mutation. And as its name suggests, that's where one or more nucleotides is left out by accident when DNA replicates. Another kind is called insertion, where one or more are added by accident when DNA mutates. And there are more exotic kinds too, where whole sections of DNA can flip around and stitch themselves backwards into the rest of the DNA, and that's called an inversion. And there are things called gene duplications, where a gene, which is the code in DNA that tells it how to make a particular protein, or that kind of gets accidentally duplicated. And sometimes you can duplicate the whole genomic complement, and that's called genome duplication. So I just want to familiarize you with the different kinds of mutations that occur. And these occur in all branches of life, from viruses and bacteria up to fish and birds and people. Now, what can mutations do? Now, in order to understand it, I'm going to use a very well understood example, which is not a virus, but it is very medically important, like the virus. And that is the case of malaria. As most of you know, malaria is transmitted by mosquito, but the malaria itself is a single-celled organism called Plasmodium falciparum. And when a mosquito bites a person, this single-celled organism gets into the person's bloodstream eventually, eats red blood cells, and uses that energy and material to make copies of itself. And so that can oftentimes lead to death. And we know that it's a big public health problem, kills maybe 500,000 people per year. And it seems to have been around for about 10,000 years or so. And it turns out that malaria is, these days, it's mostly in tropical regions, most intensely in Central Africa. And curiously, that is also where a particular mutation in the human genome is found very often. And it's called the sickle cell gene. The sickle cell gene. I'm sure you've all heard of it. It can lead to sickle cell anemia in persons that have 2 copies of that gene, one from their mom and one from their dad. But if the person has 1 copy of the sickle cell gene and 1 normal hemoglobin gene, then they have a measure of resistance to malaria and don't really have much illness because of the sickle. So a mutation in humanity has conferred on it a bit of resistance to this infectious disease. But let's ask ourselves, what is that mutation? How does it work? Well, it turns out that the sickle cell gene is a gene, oh, I'm sorry, I went the wrong way. It's a gene that codes for part of the protein called hemoglobin, which you know is the protein that binds oxygen. It's found in red blood cells, and it binds oxygen and carries it from the lungs to the tissues. So what is the mutation in hemoglobin that causes it to be the sickle version of hemoglobin? This is the molecular structure of the predominant form, the normal form of hemoglobin. Well, here is the sequence of units called amino acid residues that comprise regular hemoglobin. And as you can see, in one part of it, there's over 140, and the other one, there's around 145. And there's 2 copies of each of these subunits in a normal hemoglobin. And altogether, then, there are close to 300 different residues, amino acid residues that comprise hemoglobin. Well, what changes? What comprises sickle hemoglobin? There's to get to sickle hemoglobin, there is one crummy little amino acid change. At the number 6 position of the beta chain, you substitute a valine for a glutamic acid. So sickle hemoglobin looks just like normal hemoglobin, but there are these tiny little changes in one kind of chain of hemoglobin, where the yellow dots, where I put the yellow dots. So the basic structure of hemoglobin is totally unchanged. There's this minor, tiny little change in the hemoglobin itself. And it turns out those tiny little changes allow bumps to form on the surface of one hemoglobin, and they fit into depressions that were already there in normal hemoglobin. And this is an oily patch, and this too is an oily patch too, so they fit together nicely. But because hemoglobin has two-fold symmetry, this guy also has an oily patch, which fits into this oily depression, and this one does too. And so they polymerize, they form long, long chains associated with each other. And here's an electron micrograph of a sickle cell in which the hemoglobin has all polymerized, and it forms these very long chains, and that pushes on the cell membrane and distends it, and that's what causes sickle cell disease. So my point is that this response to an infectious disease is just a tiny change in a pre-existing molecular machine, hemoglobin. Well, there are other mutations too that have arisen in the human population and which confer a bit of resistance to malaria. We don't have time to go through them all, but it turns out all of them break genes. Either they break hemoglobin or a portion of the hemoglobin, or they break other kinds of genes. And for various reasons, breaking these genes gives a measure of resistance to malaria. Okay, so the point of this is, number one, to show that when mutation does change a protein and you get this beneficial effect, it's oftentimes this tiny little change in a pre-existing protein. And more often when it mutates, it breaks the protein that has suffered the mutation. But we've seen the COVID virus and Bacteriophage T4 viruses, you know, they have all sorts of functioning complex machinery. How did that arise? How did that evolve? Well, that's been speculated about for a long time. And here's an example from a recent review on the origin of viruses. I just want to say, quote this part: viruses are ubiquitous parasites. It is widely accepted that viruses are polyphyletic, which means they arose independently from each other, different types of viruses. But a consensus scenario for their ultimate origins is still lacking. In other words, nobody has a clue where viruses came from. It's much like the origin of life. There's lots of speculation, but nobody has the foggiest idea where they came from. So those machines, the bacteriophage machines and the machines that COVID deploys, are unexplained. And how about the coronaviruses? Some people say that, well, did it mutate and arise just in the past, 20 or 30 years? Well, no, it turns out that one estimate is that it's been around for perhaps hundreds of millions of years. That is, the basic structure has been there all along, and it's just undergone this minor tinkering that I've shown examples of with hemoglobin. So what makes it dangerous? Why did the COVID-19 suddenly pop up now and start to cause havoc? Well, it turns out that tiny changes in those viruses can be medically important. They don't really affect the structure of the virus or how it works very much, but they can affect its medical importance. Here's that slide again of how a virus binding to a receptor on a cell. And what if this is not complementary, however, to the receptor? What if the surface of the virus doesn't bind to it? Let's look at the next slide here. Here's that slide again of two proteins trying to bind each other. But now you'll notice that I colored this dot blue. So when it tries to bind to this oops, I'm sorry. I'm stepping on my slides here. When it tries to bind to the other protein, now you'll have this positive charge right next to another positive charge. And we know that positive charges repel, so they won't bind each other, okay? Even if you have the other stuff, these are repelling each other and they won't bind. But what if a mutation comes along here and changes this blue residue, which represents a positive charge, to one that has a negative charge? Now, when it tries to bind that protein, now the negative charge can bind to the positive charge, and the two proteins can stick to each other. So the point is that you can tinker around the edges of a virus, and it can change the medical importance of the virus to humans if it lucks upon a surface that can bind to a protein, to a human protein. But another large point is that new viruses are not evolving by Darwinian means in our experience. For all anyone knows, we know nothing about how viruses arose. We don't even know how classes of viruses arose. But it's very important that we know that they can become more or less medically important by little changes that can affect their ability to bind to human cells. Okay, I have a bit more to talk about, but I'm going to wind it up right now because I'm going on a little longer. But I just want to say that other considerations about how cells and molecules and life evolves can be found in my recent book, Darwin Devolves. And the gist of the book is that rather than evolving, Darwin's mechanism of random mutation and natural selection overwhelmingly works by breaking genes that were already there, as in the case of malaria, and selecting them because they might have positive effects in dire circumstances. Okay, so thanks very much for listening to me, and I will stop sharing my screen. Whoops. Stop share, and I will be happy if anybody has any questions to try to address them.
Mike, thanks ever so much for that talk. That was really interesting. Thank you. For the people following on YouTube, now is the time to type something into the YouTube chat box if you've got questions that you want to put to Mike, and I will put those my end. We have had some questions in advance by email, Mike, so I'll start with one of those if you don't mind. A lot is being said at the moment in the press and articles concerning the search for a vaccine. Do you have anything to say that might shed light or provide some information on the possibility of a vaccine in respect of coronavirus?
Well, I don't have any inside information, but as you know, our immune system does a terrific job trying to fend off these microscopic and molecular invaders. And but one way it works is that when it sees a new protein or a new molecule that it hasn't seen before, it suspects that it's a foreign substance like a virus or bacteria, and it tries to make antibodies against it, which will trigger further action. But that process can take time. So in the meantime, if the virus is stronger than your immune system, it might have enough time to replicate and destroy your lung tissue and all sorts of bad stuff. And a vaccine takes a piece of the virus, or maybe sometimes even the whole virus, which has been inactivated. and tries to select one that will, a piece that'll be most, will be good for your immune system to recognize, and injects that or puts that into a person, and that little piece causes an immune reaction from your immune system, but it does not harm you because it's only a piece of the big machine. And then if you encounter the live dangerous virus later, then your immune system is already primed to go and fight it, and so it has a much better chance of defeating the virus than otherwise. From what I read, is probably what you read, is that virus vaccines are on the way, maybe by fall, maybe by early next year. But that's pretty much how they work.
Thank you. That leads to a question on the YouTube channel, which is this. Do you think one day human genetic engineers could significantly improve the human immune system to make humans much more resistant to a wide variety of disease? That also is followed up with a follow-up question of whether you have any thoughts on the implications.
That's a very good question. I think that it's entirely possible that human scientists will figure out good ways to introduce vaccines for a wider range of organisms or viruses and pathogenic bacteria. But I don't think that human scientists will significantly improve the actual immune system itself. The immune system is extraordinarily complex, and it has to be kept within very tight borders, because if it's too lax, then a person gets infected by bugs and they eat him, and that's the end of that. But even if it's too vigilant, that can lead to autoimmune disease. The immune system starts attacking you rather than the viruses. So that's the double-edged sword. The system is ever vigilant for foreigners, but it has to know what is foreign versus what is yourself. And I am quite skeptical that there can be significant changes to the immune system itself to pump it up without causing more problems than it solves.
Thank you. Next question is this. There's two questions, but they're linked, so I'll ask them both. The first is this. If you think coronaviruses are the result of design, can you talk about the implications given that coronaviruses are designed to harm and even kill us?
Sure.
The linked question to that is, we think of viruses as being harmful and pathogenic. Is that an oversimplification? And are there circumstances where viruses may be important to the flourishing of life?
Yes, excellent questions. Yeah, I certainly do think the coronavirus was designed, that Bacteriophage T4 was designed, and life in general, every aspect of life was designed to a very deep level. There is contingency in life, but to a very deep level, life required purposeful design. Now, coronavirus hurts us. And so does this mean the designer is evil and likes to see people suffer? No, no, it doesn't necessarily mean that, because we live in a very big world and a very complex ecosystem. And as the second question implies, it may be the case, and most people think it in fact is the case, that viruses have roles in the biosphere, some of which are very necessary for other life to exist. And if viruses en masse departed this earth, then other life would quickly die out as well. And it's just that as I wrote in an article a little while ago, that just like water is good, we know water is good and rain is good too. But if you're caught in a hurricane at sea, you might have reason to ask yourself, why me? Why is this happening? Well, in the same way, you can think that storms arise in the virus sphere, so that every now and again, some parasite mutates so that, now it can stick to this protein and deploy its already available machinery. But that doesn't mean that the overall life on Earth isn't good or isn't good. Let me add that in my view, I'm no philosopher or anything, but in my view, the fact that some viruses sometimes and some bacteria sometimes are dangerous and sicken humans is no more philosophically important than the fact that, say, lions sometimes eat people when they go out and take a walk in the woods or something, or that snakes sometimes bite people when they get trodden upon in the woods. Snakes and lions, most people would agree, are great parts of nature, interesting parts, and they have roles that are not, directly concerned with humans. They might accidentally come upon a human and with bad results, but it's not that they are targeted to humans. And I think one can look at viruses and bacteria in the same way.
Thank you. I'll give you a warning that the question after this is going to touch on whether coronavirus has been man-made. But the first question before we get to that is this: do you know of any example of a novel protein-to-protein binding site evolving?
Well, the one I talked about, sickle cell, that hemoglobin itself, normal hemoglobin, doesn't bind to itself, but with that little change in normal hemoglobin, it can then associate with itself. But as I emphasized, it's a tiny change. It doesn't do anything really constructive. It does have a benefit in that it helps fend off malaria, but it also exacts a really nasty price because people with 2 copies of the sickle cell gene suffer from sickle cell anemia, and that's very bad too. As I write in my books, I think as a rule, developing a new protein binding site is extremely difficult for a Darwinian evolutionary process. But I don't want to say it could never happen. But as you get up in complexity from merely one crummy, protein binding site into these complex structures such as Bacteriophage T4 and even things much less than that, then in my view, the likelihood of Darwinian processes accounting for that quickly goes to 0.
Okay, and now the question I promised. It revolves around the suggestion that coronavirus may have been man-made, or scientists may have had a say in it. You talked about where a virus, or we don't know where viruses come from. The first part of the question is this: do you think humans could create a virus or viruses either from scratch or by way of modification from a pre-existing virus?
Okay, answering that question, yeah, I do think humans could make more potent viruses. Gee whiz, I go to the movies, I see that happening all the time, just like you folks do. But yeah, the viruses are relatively simple. And if you splice this piece of that virus to that piece of the other one and so on, you don't have to know all the details to make it really, really nasty. And so that's why for a long time, such biological research was forbidden. I think it might go on these days. So yeah, that's something to worry about, something countries should keep an eye on, a sharp eye on. Now, with the coronaviruses themselves, as I said, have been around a long time. So nobody invented a coronavirus. Did anybody tweak a pre-existing coronavirus and make it more powerful? And maybe that's the origin of what we find here. And I'm very much inclined against that. You can never rule out design. As I've written in some of my books, even, intelligent agents can make things intentionally to look like they were accidents, like any murderer who wants to pass off his killing as an accident tries to make that design look like it was an accident. So you can never say something wasn't designed. The question is, do we have any positive evidence to lead us to that conclusion? And again, I don't have any more information than is available in newspapers and journals and so on. And from what I read, that's been looked into, and a lot of people are unconvinced. There are pockets of people here and there that talk up the possibility. But right now, I have no reason to think that the virus was designed.
Okay, thank you. Next question. You shared previously that mutations in the malaria virus shows evolution does not happen observed. Could you explain? You said observation trumps theory, as you said. I'm reading word for word there.
Okay, I think the questioner is alluding to an argument I made in my second book, The Edge of Evolution, where I said a couple of things. I'm not sure which he has in mind, but the argument in that book was mostly that chloroquine, a drug that kills malaria, was effective for a very, very long time, and then malaria developed resistance to it. But with other drugs, the malaria developed resistance much, much, much more easily. So chloroquine was a difficult drug for the malarial single-celled parasite to develop resistance to. And it was discovered 5-6 years ago that the reason is that the cell has to have not one mutation, but two specific mutations in two different spots before it gets any resistance to chloroquine. So if the genome, if the number of parts in DNA is around 1 billion or 10 to the 9th, then the odds of getting this first mutation are 1 in 1 billion. And the odds of getting the second mutation are 1 in 1 billion too. And that's 1 in 1 billion times 1 in 1 billion. And that's an enormous number. But there are lots and lots of malarial cells in the world, an astronomical number. And I used that data to make the argument that getting two coordinated mutations is quickly getting beyond Darwinian abilities. And if you need more than that, then it gets to be impossible. So even with an enormous number of cells, the malaria was only able to come up with 2 crummy point mutations in a particular protein. Nothing more exotic than that, no new molecular machines or something. So the point was, to build these coordinated molecular machines like, say, Bacteriophage T4 and other things that fill the molecular level of life is certainly impossible by Darwinian means.
There's a related question to that, which you've answered partly, but it may be referring to research that I know has come out since you wrote the book, Age of Evolution, and it's this. Has any progress been made in understanding the mechanism of chloroquine resistance in Plasmodium falciparum?
Yes, there has. It's lots of progress. The mechanism is pretty well understood. There's a protein that, well, the malaria is a single-celled organism, just one cell, and it's got inside it has a little digestive compartment, a food vacuole, kind of like its stomach. And in the membrane of the stomach, it's got a protein that acts as a pump. And the pump can pump things out of the stomach. And it turns out that chloroquine accumulates in that digestive vacuole for technical reason. And usually that kills the parasite. But the mutations that allow resistance are in that pump. And they allow the pump to pump out chloroquine, which otherwise would be stuck there. And so since they can pump it out, they don't suffer the problems that sensitive cells have. And it turns out that it's the reason it can't pump it in the first place, and the mutant version can pump the chloroquine, is because chloroquine has some positive charges on it, kind of like the cartoon proteins that I showed today, and or negative charges on it, positive. And the protein then undergoes mutations that increase its negative charge so that it can now bind to the chloroquine and pump it out. But nonetheless, just like the examples I emphasized today, there are only 2 amino acid changes in the malaria that are required for it to be resistant. And they didn't build any new molecular machines. They just made very small changes in a pre-existing protein.
Okay, coming back to coronavirus, a specific question on coronavirus is this. The question is, I have seen a scientific opinion that having sufficient levels of vitamin D in the body can help minimize the intensity of COVID-19 if infected. Do you think there is merit in this?
Frankly, I'm not sure. I've read a lot of things that propose one approach versus another. I haven't heard much on vitamin D, but and again, I have no inside information. I'm sure there are teams of scientists working on all aspects of this. So we'll just have to see.
Okay, and the next one, when COVID-19 first hit the news, the accusation was made that the virus had jumped from bats to humans through a Chinese wet market. Would you comment on the linkage to other organisms?
Yes, coronavirus can infect not only people, but other organisms too, including bats and pangolins and other exotic creatures. And so the idea was that if a human gets too close to some animals, then the virus in it, if there was a mutant in it and it can bind to a protein in the human, then if the bat spits on you or some such thing, you might catch that virus, and the virus will have jumped the species barrier and start infecting humans instead of bats. In order to try to track these things down, what scientists do is to sequence the exact order of subunits in the genome of the coronavirus or whatever virus you're thinking of, and compare it to the sequence of viruses they might find in other organisms. Say they might find a coronavirus in a bat and they'll sequence that. They might find one in a human, they'll sequence that. They might find one in a pangolin, sequence that. Find one in a fish in a wet market somewhere and sequence that. And then they compare the sequence and see which is the closest, which has the fewest differences between the human one and the other possibilities. And they try to link these things up as in a puzzle and figure out how the virus got the ability to infect the human community. And again, as far as I know, that's still up in the air. So these things are a whole lot more difficult and to solve than most people appreciate, and we still haven't done it yet.
Returning to the question of whether there was any human influence on the development of coronavirus, the question is this: is it possible for viruses, and I'll be specific, the coronavirus, would it be possible for that to be made harmful as a result of human or synthetic factors, influences? You talked about the murder and the accident. You may have been talking about that in theory. In practice, do we have the scientific knowledge and skill to bring about those changes?
Well, yes and no. We know that where dangerous viruses are, and we have ways to alter their DNA or RNA sequences in ways that we think will make them more dangerous. And I think militaries around the world have sometimes done this sort of thing. But very often, the changes that are made don't help or don't make it worse, don't achieve the goal of the people doing the project. Because even tiny viruses are a whole lot more complicated than we can easily understand. So one technique is oftentimes to make a whole bunch of viruses with a whole bunch of sequences, and then try to select a virus that will be more harmful than others. So one might make a bunch of viruses and inject them into some, say, poor bunny rabbit or something, and see which one reproduces the most and which can infect other bunny rabbits in nearby cages the most. And so by a combination of human guesses and selection of these designed objects, probably some nasty things can be made.
Okay. Returning to the subject of mutations, the question is, you have said mutations are occasionally beneficial, but then they usually break genes. Can you explain the exception, that being the beneficial mutations that don't break or just duplicate genes?
Yeah, sure. The ones that don't break genes usually just, affect them neutrally. They do not confer on them new and complex properties. For example, there are some hemoglobin mutants that, like sickle hemoglobin, confer a bit of resistance to malaria, but do not aggregate and form those needle-like structures that sickle does. And the hemoglobin can work just fine, and it doesn't cause problems when from the mutation. So that's a mutation that doesn't break a gene, but it certainly isn't making one either. It can help to duplicate genes, especially in bacteria. Scientists can grow a bacterium sometimes in the presence of an unusual food, one that it usually doesn't metabolize. And some genes code for proteins that can produce, and usually produce in very small quantities, enzymes which can help to digest that unusual food product. And over time, scientists who culture these bacteria might notice that they're growing faster or to a higher density. And when they go back and look, they find that that single gene that helped them with that unusual substrate have duplicated, undergone gene duplication, like I pointed out in the table. And sometimes more than once, maybe 10 times or 20 times. And so they produced 10 or 20 times the amount of that enzyme, and that is beneficial. But again, that's nothing new. You're just essentially copying the same thing multiple times. So those things do happen, but as I write in my new book, Darwin Devolves, in our experience, most helpful mutations turn out to be ones that degrade or break pre-existing genes. They do not make new or complex molecular machinery.
Okay. Next question is this. I've understood that proteins are highly improbable to be made by accident. How is it that mutations on proteins are relatively common?
Well, that's a good question. So yes, the original statement, yeah, that's certainly true. To get a protein that works and does something like biological proteins is expected to be very, very unlikely, because like the hemoglobin that I talked about, they consist of, you know, a hundred or hundreds of particular amino acid residues, and the very sequence of those, which one you put next to which, is critical for allowing the protein to do its job. And this has been likened a lot to, you know, coming up with a couple of English sentences in a row that convey some coherent message to it. Most combinations of 200 letters will not give you anything that makes sense in English. It's very rare. And it's the same thing with these proteins. It's very hard to come up with it. But once you have it, like the hemoglobin, getting a mutation in it is not that hard. All you have to do is make a mistake. Making mistakes, as we know from our own lives, is much, much easier than constructing things. And the cell, again, although it's very good at what it does, occasionally it makes mistakes too. So for example, in the sickle mutation, one can think that, 10,000 years ago, a baby was born in which during DNA replication, the machinery made a mistake when it was making a copy of its mom's or its dad's hemoglobin gene and substituted a code to put in valine for glutamic acid at that particular position. And then when the baby was born, all of a sudden, it was not getting sick like its playmates were. A lot of kids will get sick with malaria in various regions of the world, but this Sickle Eve, I called her in Edge of Evolution, she was resistant. And so she had a great chance to go on and grow up and marry and have kids who might inherit a copy of that gene too. And then they would go on and have kids, and eventually the sickle mutation would spread widely. So once you've got a functioning protein, to have a mutation occur in it, all has to happen is a mistake be made by the replicating machinery, and that's where they come from.
Okay, thank you. We've got time for one more question. Just before I ask it, I should say there are a few questions that we haven't had time to get round to. Although I'm not going to get to ask those questions to you this evening, I will keep the questions. I've saved them. And if we get the opportunity, I will try to ensure that next week's speaker gets to answer those questions. So it's an incentive, at least, for the 3 people whose questions I haven't got to listen next week. But the final question tonight then, Mike, is this, if I can find it. Can you clarify what you mean by viruses don't evolve by Darwinian means? As from what I understand, they undergo mutations and are subject to natural selection.
They do. That's exactly right. They do. In my remarks, I was confining that assertion to the general structure of the virus, the initial structure of the virus. We do not, as the article I quoted says, scientists do not know where viruses come from. And that means that the original viruses that have their genes that give them their molecular machinery are unexplained. What we do know is that Darwinian processes can produce these very small changes, maybe in the surface of a protein, that lets it bind to something better, just like, say, sickle hemoglobin binds to itself. But if you're thinking about something like, let's think about Bacteriophage T4, the legs of it, the shaft, the needle, the ability to curl up its genome in the head of it, all of these things working together for one purpose of essentially having an automated syringe shoot the DNA into the bacteria. That's a coordinated, very complex machine. And as for reasons I talk about in my newest book, there's virtually no chance that that could be produced by Darwinian evolution. That's what I meant by that statement. I certainly didn't mean to say that, tiny changes here and there couldn't happen and you can get a little bit of variation around the edges. But the basic structure of those, and even some non-basic structures of them, are beyond Darwinian explanation.
Mike, thank you ever so much. I'm afraid we've run out of time. We could have gone on a lot longer, I think, but that's it for this evening. So thank you from me, and I'll take this opportunity now to hand back to David Galloway for some closing remarks. Thank you.
Okay, thank you, David, and thank you, Mike. Thanks both of you for getting this series of webinars off to a fantastic start. Mike, thanks very much, particularly for your talk, covering the history, a bit of the biochemical, to see how you will certainly get the philosophers chewing and the skeptics chewing when you made a very clear statement in an answer to one of the questions about your belief that viruses are products of design. And so that was why we asked the very question in the banner headline. And I think it's a very interesting question to explore further. So the only thing for me to say now is to remind everyone that next Thursday, not Friday, next Thursday, the 23rd, at 8 p.m. UK time, 3 p.m. Eastern Daylight Time, we will host the next webinar, and that will feature one of our own committee members, Dr. Jonathan McClatchy. Jonathan has some academic background in forensic science, in evolutionary biology, and in molecular biology, and he's going to be dealing with the topic of the molecular biology of coronaviruses. So, Mike, once again, we're deeply indebted to you for taking the time and the energy and the effort in putting this together. And let me thank you once again, and thank everyone who has been involved in watching and in participating and making comments and asking questions, and to David for posing those questions. And we look forward to seeing everyone next Thursday. So thank you very much and good evening from the UK.